So thank you, everyone. This panel discussion is on the gene therapy, the outlook for 2018. And I think, as we heard this morning in the state of industry and in the previous panel, 2017 was definitely an inflection year for gene therapy, from our first approvals, both on the CAR-T products as well as for the first in vivo gene therapy in Luxturna, as well as a number of really exciting clinical results. A lot of promise in the field that we're seeing in 2017, both the progress we're making against hemophilia and beta thalassemia, sickle cell anemia, as well as our first in vivo gene-edited clinical trial starting this year. So lots of excitement in 2017, and I think we're really geared up to have continued excitement and a really promising outlook in 2018. So hopefully I'm not giving spoilers for the panel discussion that the outlook is good, but to tell us a little bit more about that in the details is our panel. So to start off, I'm going to ask each panel member to introduce themselves, their company, and their experience in gene therapy. Go ahead.
Good morning. My name is Deepak Srivastava. I'm actually here wearing multiple hats, I imagine. I serve as president of the Gladstone Institutes here in San Francisco. I'm the vice president of the International Society for Stem Cell Research, and I'm co-founder of a new startup company in the Bay Area called Tenaya Therapeutics that's focused on heart failure, and in particular a gene therapy approach to creating new muscle cells in the heart, and that's probably the reason that I'm here with you today, this morning. And so our laboratory over the last many years has identified a combination of genes that are normally present in a developing embryo that when we reintroduce into an adult damaged heart are sufficient to convert non-muscle cells in the heart, which are abundant, into brand new beating heart muscle cells. And so essentially we're able to regenerate damaged hearts from the cells that are already existing and are resident within the organ. So it's not a cell-based therapy, but rather a gene therapy to give the right cues. And we think this approach is not unique to the heart, but could be a paradigm for multiple different organs. And there are multiple groups around the world now trying this similar approach with different cocktails of key regulatory genes that could induce the cell conversion. But all of those then require an effective delivery of the gene products. And so we've explored a number of different viral vectors. Adeno-associated virus may be the most viable, but is limited by packaging, of course. And so I think one of the key issues going forward is, from a regulatory standpoint also, and a production standpoint, is how viable is a combination of vectors for a single therapy. How viable will that be, and can that be taken forward towards clinical trials? Safety issues, of course, and issues around really delivery, because you have to, in this situation, you have to get high enough levels of gene expression to wholesale convert a cell from one cell type to another, which it doesn't want to do. And so non-integrating lentiviruses probably don't give you enough, high enough levels. And so really local delivery, high enough levels of expression, production, and numbers of vectors, I think, are some of the key challenges we're facing.
Hi, good morning. I'm Amber Salzman. I'm CEO of Adverum Biotechnologies. We have industry-leading capabilities in terms of novel vector development and process development. And we also have a very robust pipeline, and we're excited that our lead indication in alpha-1 antitrypsin deficiency started dosing at the end of last year. Very exciting for the patients. And we have 2 INDs planned to be submitted second half of this year. One in wet AMD, and that's where we have very promising efficacy data in the non-human primate model. We seem to have unique capabilities in terms of delivering our vector to the eye with an intravitreal approach, so it doesn't require the subretinal surgery, and it's standard of care in terms of wet AMD, with the exception that you have the long durability. We also have an indication in hereditary angioedema. Again, really promising data in the mouse model of the disease, and we're excited to be able to submit these INDs in the second half of this year.
Good morning. My name is Bill Lundberg. I'm the Chief Scientific Officer of CRISPR Therapeutics, and I'd like to ask ARM for inviting me here, thank you, to share our story. CRISPR was founded in 2013 on the foundational discoveries of Emmanuel Charpentier and Jennifer Doudna, and the foundational observation that you can take this bacterial system and re-engineer it or exploit it to gene edit in any cell type, and in every cell type. And when we were founded with a clear mission to create transformative gene-based medicines for serious diseases. So we've had an initial focus on taking the technology and translating it into a therapeutic approach. 4 comments about the company. The first is that focus has led us to being able to say that we've filed multiple clinical trial applications now at the end of last year to bring CRISPR-based gene editing into the clinic for patients. The initial indication is beta thalassemia with sickle cell disease to follow this year, and bringing it into the US later this year as well. The second is that we have a robust partnership with Vertex, who opted in just a few weeks ago. We're thrilled to have them as a partner. As all of you know, they've just done a tremendous job discovering and developing therapies for cystic fibrosis and commercializing them. And so we have a real opportunity to partner with them and learn from them in our journey. The third is Immuno Oncology, and I would say from this session we just had, enough said, but I'll make a couple of points here. When I was clinically active and seeing patients, I couldn't even imagine that we would be getting the rates of remission and response rates in hematologic malignancies, particularly molecular remissions in through BCMA and multiple myeloma. It's just tremendous. But the challenges that we see in our hypothesis is really around creating these therapies for patients, and the opportunity is to use a CRISPR-based platform and technology to create allogeneic or off-the-shelf cells for patients. And then lastly, we, together with Pharmaceuticals, have created a brand new company, Casivia Therapeutics, with an entire portfolio of opportunities. And there's much that hasn't yet been said about Casivia, but we'll be hearing a lot about them in the coming months and the programs they're moving forward with.
Great. My name is Geoff Nichol. I'm the Chief Medical Officer at BioMarin, a company that I'm sure many of you will be aware of. It's been around for about 20 years and has developed a range of treatments for rare diseases, very much a pioneering company in the rare disease space. And chief in its current development pipeline is an AAV5 delivered factor VIII cDNA for the treatment of hemophilia A. This has completed phase 1 and phase 2 development and is currently in phase 3 clinical trials. This is a new platform for BioMarin, and one that we continue to invest heavily in to look at other opportunities in gene therapy, but we're very, very pleased to be currently a major leader in the development of gene therapies for hemophilia.
Good morning. I'm Sandy Macrae. I'm the CEO of Sangamo Therapeutics. We're known as the zinc finger company, but we really are a company whose fundamental premise is to take groundbreaking science and convert it into medicines for important diseases. So we have 5 clinical programs open in hemophilia, MPS 1, MPS 2, and recently in thalassemia. We were the company that did the first in vivo genome editing last year, which was a moment of great excitement and also huge responsibility, where for the first time we change a bit of the genetic, a bit of the DNA of an individual. And I think that is going to be the first in many such advances that many of the companies here will take forward in the coming year. So we have our clinical programs. We have a fascinating middle pipeline that involves both oncology and CAR-T, and where we can edit T cells based on work we did with HIV for many years. We have about 100-odd patients in HIV that have had their T cells edited and have shown the safety and effectiveness of that treatment. And then with our gene transcription control, we're looking at tau for Alzheimer's, and we recently announced a deal, a partnership with Pfizer to look at C9orf, which is thought to be involved in ALS, and that's another example of taking really cutting-edge science and matching it with the capabilities of a pharma company to address an important medical disease. And finally, we have our platform. And one of the most wonderful things about this job has been discovering just how good the platform is. And one of the things I think would be interesting in this panel is to understand how do you look at the different platforms? How does one understand the differences? Because as we all move from this being individual diseases with individual companies to a world where there's many companies in gene therapy and gene editing, we need to start to develop a lexicon and a way of describing and comparing what we all see and do, so as all of you can understand the advantages and sometimes disadvantages of the different technologies.
All right. Thanks to the panel for those introductions. It sets up a great starting point for the conversations. I think we wanted to start with a little bit about the clinical progress, both the exciting results of 2017 and a bit of the outlook for what we're going to see in 2018. So Geoff, did you want to start with some of the hemophilia results that BioMarin has?
Yes, I mean, hemophilia has been traditionally a gene therapy target for several decades now, and it's easy to understand why that would be. AAV tends naturally to go to the liver, which is where the clotting factors are primarily made. You only need to produce a relatively small amount of protein at a steady rate in order to have a profound effect on the disease. And the level of expression that you need can vary from small to quite large with a wide therapeutic index and still have a profound effect on the disease. So it's always been attractive. And with very promising early data with hemophilia B from Amit Nathwani and others, including animal data, here we are seeing human beings for several years producing factor IX. And so one would hope that factor VIII and hemophilia A, which is a much more common form of hemophilia, would be just as easy to deal with. Well, it turns out it hasn't traditionally been so. For a start, the factor VIII construct itself is very large. It's very difficult to actually fit into an AAV vector. You need to remove the B domain. Mercifully, you can remove that and retain the activity in order to fit it in. It's pretty immunogenic in the animal species that you need to test it in. So it seems to have been very poor at expressing in a lot of animal settings, even though the low levels that have been expressed have been very effective. I think at the time that the very first human studies were happening, people would have cracked the champagne if they could have got 5% of normal expression. But in fact, in the very first phase 1 study with the construct, which is currently being carried forward by BioMarin, essentially knocked the leather off the ball with essentially normal expression levels in human beings. And we've continued to move forward with our program. We have chosen 2 doses, one which essentially gets the majority of patients into the normal range for factor VIII, and another dose which gets the majority of subjects either into the low normal or the high mild range. And those two doses are being taken into an ongoing phase 3 program. We look forward to progress with that program. All of the subjects in the phase 1 and phase 2 programs who've achieved therapeutic levels of factor VIII have stopped bleeding. So this is a transformational product. And it's becoming an expanding field. We very much look forward to data from Sangamo and from Spark in hemophilia A, and we see Spark Pfizer as well as Unicure moving forward into phase 3 with hemophilia B. So a huge area. We've invested enormously in it. I think there needs to be a great deal of education as to what gene therapy means and how transformative it can be, and clearly a lot of investment and a lot more work on manufacturing. I'm sure we'll be talking about that a little later on. And of course, very challenging in terms of reimbursement. One treatment more or less cures the disease, saves an enormous amount of money, but how do you get reimbursement for that?
Yeah, and I think that's it's a great point, and also a good segue into the concept of gene therapy and the excitement going on around gene-edited gene therapy as well. So if the panel could talk a little bit about what they consider to be the similarities and differences both around mechanism, upcoming clinical, and some of the platforms required, that would be great. Whoever wants to go first.
Sure. So obviously both of these are gene-based medicines, and they take very different approaches. One is the transgenic or transactivation or expression of a gene that's introduced. The other is modification of the endogenous DNA that's already in the cell. The differences have to do with complexities around gene regulation and what gets expressed, the complexities around gene vector delivery and gene therapy. The similarities have to do with how do you get your tools in there in the first place. And I think even among the different gene editing platforms, there are different approaches to introducing the molecular scissors in, even between a zinc finger approach and a CRISPR approach, which is a protein-based therapeutic.
But I think it's interesting to see the intersection between the gene editing and gene therapy. So while gene therapy can only deliver the gene, when I say only, it's still greatly beneficial, there are some diseases where you actually have to edit the disease because of the pathology. However, if you're looking at having to do that kind of editing in vivo, so you have to get into the cell, you still need some kind of technology to deliver, whether it's CRISPR or zinc finger, to actually get into the cell. And there's different approaches being entertained, and this is where the AAV, for example, gene therapy approach can be quite helpful. So, I mean, for example, that's probably what led to the collaboration we have with Editas. We have unique vectors that have a great way to get into the eye intravitreally, so if they're trying to get their technology in, they'd be interested at using our vectors to get there. So again, it's a nice intersection between gene editing and gene therapy, depending upon where you actually have to implement that type of technology.
It's probably worth considering that if you're able to get the machinery and the tools in for CRISPR-Cas9, the opportunities are there not just for gene editing with that machinery, but epigenetic programming. So if you want to control a cell and its gene expression, and particularly in cellular reprogramming, it's something we think about a lot. There are ways now that you can turn on or off genes at specific loci using the CRISPR-based guide strands, and there's a lot of potential there as well.
So like most things, it's simple and complicated. So the simple answer is eventually the right thing to do is to edit the disease and correct the mutation or the genetic error, and bring the patient back into that they do no longer have the disease. But at the moment, the thing that's most difficult is delivering. And so we have to use a range of technologies. And at Sangamo, we decided that we would be agnostic to the form of treatment and match it to the disease. So for hemophilia, it was logical, and Geoff used to work at Sangamo and deserves credit for our programs in hemophilia. It's a logical thing to use for hemophilia A. For some of the rarer diseases where there's consequences to the patient having the disease in early childhood, gene editing seems to be the right way to go forward, because you give them a solution that will last for life. It's yet to be understood whether gene therapy will be long-lasting in animals. It's perhaps 10 years, maybe 15 years that we can see data on. But for things where you're going into a child or an infant, we feel gene or genome editing is the right way forward. But a company that's able to do gene editing is also a company that's going to be able to do gene therapy. So we look at it as a suite of capabilities that we apply to the right medical problem.
Yeah, and I think you see that a lot in the discussion in the field. When is a gene-modified cell therapy considered a gene therapy versus in vivo gene therapy? And now there are additional uses of gene editing around regenerative medicine. Did the panel want to talk about any of those opportunities or development areas?
Take this.
In terms of regenerative medicine therapies, gene editing, you could consider something like Duchenne's muscular dystrophy, even a regenerative problem. You've essentially, those children with that disease have lost muscle, skeletal muscle, and it's progressive. They start off normal and they get worse. Little boys get worse as they get older, so they're degenerating their muscle. And there have been approaches now described to use CRISPR-based editing to essentially restore some of the muscle in those patients that look very good in animals. And there's commercial activity there now to try to regenerate skeletal muscle that way.
And I'll say we at CRISPR Therapeutics find the field fascinating, and it's the intersection of a number of different discoveries and significant progress in scientific areas. One is in the field of cellular reprogramming and IPS, and we heard a little bit about that in the panel just before this. Another is the increasing facility with which not only we up here, but many, many different researchers, scientists in industry and academia can take the CRISPR toolkit and modify the cells. And the third is a better understanding of the physiology of specific diseases. I would predict that over the coming years, we'll have significant opportunities to create regenerative medicine-based therapies using the marriage of these various different strands of work.
I think there was such remarkable data presented earlier, well, not early this year, it was late last year, where patients who had EB, a terrible skin disease, where basically you don't have your outer layer of skin, and physicians were able to ex vivo correct the skin and then use cell therapy to grow it, and they were able to replace basically the skin 80% of this boy's body. It was tremendous. So to see that kind of intersection, that we can correct the protein so that you can have normal skin and then grow it, I mean, it was mind-boggling. I mean, remarkable.
Which just underlines it's all down to delivery. I think the technologies in gene editing, particularly with zinc fingers, where we can edit any nucleotide in the genome, it's now molecular engineering, it's not molecular biology. And the only thing that paces what diseases we're going for now are, can we get to that tissue? So I think you can now reliably get to the liver. Some companies like to put things in the eye. I think the CNS is going to be solved very soon with some of the AV9 type viruses. Look like they can reliably get across from an intravenous injection. And then questions like muscular dystrophies, once we have a reliable way to get to muscle, that is solvable. That disease is solvable. And it's such an exciting time in medicine to be at. If we could get reliably to the blood side of the lung, and I know, Geoff, this is something close to your heart, cystic fibrosis is addressable. And it's just a matter of investing and developing the right delivery technologies. And I think if I was looking at the investment in this field, that would be the areas that I would encourage you all to look at, is the discovery and the development of new AVs, of looking at lipid nanoparticles, and at new ways to get to perhaps skin being one of them.
Sandy, I would agree with that. I think that a number of papers have shown that you can correct, using gene editing, the Duchenne's disease mutations. Just to ask the question back, the title of our presentation is The Outlook in 2018. How far do you think we're going to get this year?
So I'd love to say that we're going to solve Duchenne this year, but we all know that the vectors aren't there for it. We have work going on in non-human primates at the moment, looking to see if we can reliably get across the blood-brain barrier in a larger species, because it's not quite appreciated that there's some kind of difference that happens when you go from mice to non-human primates. The predictability of non-human primates isn't what everyone, I think, assumes. If we can do that, we have a whole range of CNS targets that could keep my company occupied for the next, not just for 2018, but for the rest of this decade.
There are two approaches that we were excited about to address some of these things regarding delivery, both tissue specificity, things of that sort. And we face this in our work in the heart because there are great AAVs if you want to infect a cardiomyocyte. Fabulous. If you want to infect a cardiac fibroblast, which is our target for converting a cell to a myocyte, nobody's ever wanted to do that. And so there are no AAVs out there. So we have screened for novel AAVs using an error-prone AAV library that David Schaefer's lab here in Berkeley had made, where there are millions of random variants from mutations, and then you can do positive and negative selection screening to identify the one that might have tropism to your favorite cell type. And it's very species specific. But even then, you could get an AAV that can go into cell X, but it's probably not going to be that specific. And so we've been, now that there's a tremendous amount of epigenetic data on various cell types, we've begun to mine that data with machine learning techniques to try to identify synthetic enhancers that could then give you cell type specificity for your favorite cell type. And through the combination of that, we're hoping that we'll get to the point where we both get good delivery and get some specificity.
And I think what's exciting for really 2018 is that with some of the advances in either promoters so you get enough protein, or in manufacturing so you can make enough of it. I mean, that was the challenge in the past. Could you actually fix enough of the cells to make a difference in the disease? That was really what held us back for quite some time. And now we're starting to see either different vectors, different promoters, different manufacturing techniques, so we actually have a shot at correcting enough of the cells in different cell types. And that's what's so exciting in terms of when I look at 2018. And the fact that we have clinical data coming out, even if the first data coming out isn't a home run, even if it's a first base, and then you boost the promoter or you put a higher dose, at least we're on the way to really making a huge impact to patients' lives.
So on a very related topic to choosing the right delivery in the vector is the ability to manufacture it. And I know it's a challenge that's talked a lot about in the field, and I think we're making a lot of progress in the field as well on how to manufacture. Does the panel want to talk a little bit about some of the challenges and some of the progress they've seen and expect in 2018?
Yes, I guess having made the transition into phase 3, that's always a huge no man's land that you have to cross in drug development, and getting a reliable process that you can increase the scale of sufficiently for large phase 3 and ultimately commercialization, it takes an enormous amount of effort. There are a couple of platforms traditionally out there, the human cell 293 platform and then the insect cell baculovirus systems. And certainly at BioMarin, we looked at both. We didn't see fundamental biological differances in terms of the AAV that each produced, but we went with the baculoviral-based insect cell system because that gave us the ability to get to scale. We can produce more vector faster per cell than with the currently available 293 approaches. I think we are feeling very comfortable that we have a process that we can take into phase 3, very comfortable with working with the regulators to move that forward. But I think it's still pretty early days for just about every delivery system, and as was addressed in the prior session, there's a long way to go. It's like the very early days of monoclonal antibodies. I guess the originators tended to sort of build their own manufacturing plants as we're doing. There was a relative lack of contract facilities out there. All of these things need to be addressed, and I'm sure will be addressed as the field grows and accumulates successes. But we are going forward and looking forward to continuing to improve the processes that we use, and I'm sure we'll see across the field significant improvements in scale, in productivity, cost of goods, and so on, as we've seen with monoclonal antibodies over the decades.
It does speak, though, to what I think is one of the biggest challenges for all of us in 2018. When I first joined this field, there was a sense that this was about showing you had an IND or even showing you proof of concept data. If you believe that you're going to be successful, if you truly believe you're going to be successful, you really have to plan for phase 3, 2-3 years in advance. And some of the delays, queues for manufacturing, can be 12 to 18 months after you deciding you need between deciding you need something and having it delivered to you. And so at Sangamo, we made a decision to both have a unique supplier relationship with Brammer Bio, where we have a dedicated suite within their facility, and also in our new Brisbane facility, we'll have our own manufacturing at GMP type standard. And it's partly to control the timelines, but it's also to control the quality and to think about having more than one option in case, occasionally bad things happen. And that professionalization and industrialization of our field I think, will be what will be new in the coming year. That it's no longer about just having a nice result at ASH, it's about having medicines that's ready for the patient and ready for launch.
Yeah, CRISPR, we're obviously not a commercial stage company, so we don't have any of the same considerations or seasoned executives. But very early on, it was obvious to us exactly what Sandy is saying, that those very few initial patients we could treat could quite likely be part of a registration package for a rare disease, for a rare indication, where you only have on the order of tens of patients' experience in your regulatory filings. And we were really quite humbled by the field of manufacturing and just how damn hard it really is to get it right. And so early on, even about a year old, we hired a leader of process development and manufacturing and invested enough time and effort to be able to screw it up a few times before we got it right. And I think that early investment in recognizing that we're not going to get it right the first time, we have to iterate, we have to and really invest time and resources, has led us to be able to get to a point where we were able to file clinical trial applications last year. In the end, you can only control probably 2 out of 3 of time, quality, and cost, and you have to pick which 2 you want to control in order to be able to be successful. But it is much more difficult than you anticipate. There's always something that comes up in the landscape of manufacturing. It's not new discovery. You don't need brilliant scientists making new Nobel Prize-worthy inventions, but you need really, really good people who understand the world of manufacturing to build out the manufacturing organization and do the work that's required to get material for clinical trials or for registration.
Yeah, and I would echo what Bill said in terms of the process development skills. So you can look at companies like BioMarin or Unicure who have their own manufacturing GMP facilities as well as process development, which is a really nice strategy. But there's other ways to go about it too, which may not require as much investment early on. So if you have the right people who can do the process, you can then set up a relationship. For example, what we do at Adverum is we have a relationship with several commercial manufacturers who have the same kind of bioreactor that we do, and they can follow our batch records. So we have process development experts who have made sure that it's a reliable, robust, scalable process, and we don't rely on the manufacturer to do the scaling, because I think that's what plagued the field to begin with, is that what happened a few years ago is you'd have an academic process where they can make it a small amount, and they'd say, okay, here, manufacturer, now make it in a big scale, big bioreactor. Things don't scale that way linearly necessarily. So our approach, for example, has been we'll make sure that we can scale the process, and then we'll work with a vendor who has a great GMP facility and knows how to use their bioreactor and can follow our batch records, and we will show up at key points of that process and make sure that they follow what they should be doing and rely on our expertise to actually make it. And that opens up the door to more manufacturers and avoids some of the problem that we've heard where there's a limitations of CMOs who can actually make AAV vector. But as mentioned earlier, once you're looking at phase 3, you really do need to think about different approaches potentially, rather than just relying on the process development experts.
Okay. So great, and I guess one last question on manufacturing, because we've been very focused on vector delivery and viral delivery. With gene editing, there's a number of people that are thinking about more direct delivery of the RNP. Are there thoughts already around the manufacturing there?
You know, I can speak from the CRISPR standpoint. You can make comments about zinc fingers. So we in part of our early investment and bringing on someone who had the expertise to help teach us, we spent a lot of time trying to understand in our lead programs, which are all manipulating cells outside the body or ex vivo therapy, do we want to introduce our CRISPR tools as an RNA molecule with a guide RNA, or as a protein, a Cas9 protein with a guide RNA? And much of that is evaluating, optimizing, iterating, and then doing it all again at scale, right? And what we've established is that for our system, for our proprietary approach, and I can't comment on how anybody else does this, the Cas9 molecule, it behaves better and it is more effective if we provide it as a protein with guide RNA. And our GMP suppliers, our backup suppliers, our process development, our GLP clinical scale manufacturing process is all consistent and robust around that notion.
I would just echo everything you said. It has to be robust. It has to be well thought through. You have to invest in manufacturing. The bit that interests all the investors, the remarkable results, is but a small part of a successful gene editing or gene therapy company.
And it's often kind of boring when you talk to investors. They're like, yeah, you know, tell me about the cool science or the big opportunity or the big numbers. We don't often get into the details of manufacturing, but it's really important. The in vivo programs require delivery in manufacturing of all the delivery components, right? Whether it's nanoparticles or viruses. And that represents an area where we at CRISPR are hoping to run in the wake of my colleagues here on the panel who are helping sort it out for us.
Bill, do you think that the recent concerns raised about the potential immunogenicity of the Cas9 protein would make it so that a RNP delivery, which is there and then degraded and gone, might be, for in vivo use, might be preferable to a gene therapy approach?
That's an important point, and many of you may have seen this publication that was that came out in an open source journal over the weekend from Matt Porteous, who's one of our founders, talked about all of us, because we're exposed to Staph aureus, it's on our skin, and because we had strep pyogenes as kids, the disease that caused strep throat, those are the sources of the common Cas9 protein. It's Cas9 from strep pyogenes, the sore throat, the strep throat bug that we use. So all of us have pre-existing immunity to it, and Matt's paper that came out just quantified that in a small subset of patients, both antibody and cellular immunity. So a fraction of us have that. I think it speaks to the importance of being able to do your editing and then not have the tools last for too long in whatever cell type. And particularly for the ex vivo approaches, we're editing in the cells outside the body, and Cas9 is basically gone by the time we reintroduce anything back into the patient. But it's an important observation.
So as a eukaryotic gene editing company, we saw that with interest. I think.
Wait, does that mean you're editing humans? Eukaryotic?
We use human tools to edit humans. It seems like a logical way to do it. Just saying. We read that paper with interest, but with some love for my fellow editing companies, it's a single paper. It's an interesting result. We need to take the time to understand it. I think one of the things that's most interesting about this field is our dependence on single patient results and the excitement you've seen at BioMarin, where everyone looks at one patient and sees where they go up or not. I think the lesson from the Spark data is let's wait and let the data mature. And I would say it's the same about this paper. Let's wait and let the data mature, because I'm sure there's three CRISPR companies. The three CRISPR companies will
We would count Casibia as being the fourth independent CRISPR company.
Okay, the four CRISPR companies will be studying this carefully, and I'm sure we'll see data that will explain it and help us all understand it.
Okay, so let's move on to the next contentious topic, which is about reimbursement. If you go downstream of manufacturing to the point where we're now commercializing these therapies, there's a lot of discussion, there's a lot of new ideas around the reimbursement picture, but I think there's also a history, particularly when you look at orphan indications, of how the field has addressed these approaches. So what are the panel's thoughts on reimbursement for 2018?
Well, I think we also have to be careful not to paint them all with the same brush, because not all gene therapies are being applied to rare diseases. So, for example, we're looking at wet AMD. That is clearly not a rare disease. There's no way we're going to be charging per eye what Luxturna can charge. But that's okay. We can still be very profitable and charge a reasonable fee. So you have to look at what's standard of care right now for that disease, and if you avoided having monthly or bi-monthly intravitreal injections, is it worth some multiple of that? So again, these numbers are just much more modest. And if you look at the overall cost of having a patient come into an office and get a shot, you can maybe have a different kind of equation when you look at reimbursement. So I think first it's really important that we don't brush them all with the same paint, with the same stroke. So that's one thing that I can't emphasize enough when it comes to gene therapy. When it comes to rare disease, some of the challenges that we're hitting in gene therapy are similar to some of the challenges that some of the rare disease companies hit. I mean, I know BioMarin had some challenges initially charging high prices for some of these rare diseases. I think they did a really nice job learning over time how to work with the reimbursement and how to deal with the co-pays that patients have to make. I think we'll have the same challenge when it comes to doing gene therapy. We have to look at the total cost of not treating versus the cost of treating. I mean, I think that's obviously a much bigger challenge in the US than in some other countries. But if you look at the cost of not treating, you know, whether it's other drugs, what they would cost, the hospital costs, if you look at some of these diseases where it will beyond just the cost of medicine or hospitals, but now they no longer can go to mainstream schools, et cetera. You have to look at it in a more complete, holistic way and work with your key constituents to roll it out. And I know that I don't think there's a simple and complex answer. I think there's only a complex answer in this space.
But I think we need to talk not about cost, but about value. And I would encourage any of you to go and look at the Spark data, the videos of the children that can now see and can now walk their way around the obstacle course. That there's a question over the cost of that medicine seems to me ridiculous. That is a medicine that brings value to patients and changes the fate of these children. And so we need to demonstrate in the industry, we need to demonstrate that value and not just bang the table about the cost. And you've got to then think, I think, as Amber's saying, about the total cost to the patient. So for hemophilia, the average cost for a hemophiliac for their factor is, I think it's $6.5 million. So if you come in with a medicine that is significantly less than that and avoids all of that cost, we brought a family in to speak to us last week, and that one of the boys was taking $2 million worth of therapy each year. Surely if we can replace that with a gene therapy medicine, that's a valuable to the patient, but it's a value to society for costs that will no longer be required of us and of the insurance agencies.
Yes, James Noble at the previous session said, you know, I'm so glad I'm not the first one who's going to have to deal with these issues. But at BioMarin, we are probably going to be one of the first to deal with these issues in hemophilia. And as Sandy's saying, you know, the present value, if you like, of just simply substituting the costs for factor therapy in a patient's lifetime is many millions. And that's probably too much for most payers to be able to pay up front. In any case, they will be concerned about just how long the therapy will last and just how effective it's going to be in each individual patient, because there is some variation in how things work. The way we look at things is to recognize the huge value that I think Sandy and others have emphasized, that a single treatment that is potentially curative can actually bring to the patient, to the healthcare system, but to also recognize that not only not all gene therapies are the same, but even in settings where there's perhaps similar reimbursement, they're probably still different. It depends very much on how big and clear the effect is, how long it lasts, and factors such as that, as well as identifiable areas where perhaps you can get some gauge of value from the cost substitution argument. And then not all payers are the same. As you were saying, they're different in Europe from here, and yet even here, for example, in hemophilia, there's the government pays a great deal for factor and for a significant proportion of patients. So we tend to take the view that there will probably be multiple models around the world, different approaches that will be taken in order to reach a reasonable agreement between the innovator who's bringing enormous value and the payers who want to capture some of that value, are willing to pay for a fair proportion of it. And so there'll be significant negotiation, and I think there'll be a need for some fancy footwork and for some creativity as we move forward. But yeah, I think the one and done approach that we're talking about here is absolutely transformative to the way these therapies will be paid for.
Okay. Thank you. So I think we've about used our time up. Are we going to have any time for questions or no? So thank you to the panel for a very informed and exciting discussion.